Hello everyone, I hope you are doing well. In my previous video lecture, I showed you how can we retrieve the coding sequence of gene of interest from ENSEMBL database. And I have already pasted here the sequence of GCK gene retrieved from the database, and you may see here first yellow sequence is showing the exon number 1, and there is in green a green portion which is showing exon 2, and then yellow portion again showing the exon 3, and so on. Now in order to in order to incorporate the SNPs into this coding sequence, first we have to split this coding sequence into the codons. As you know, a codon is a group of 3 nucleotides, so we will manually split this coding sequence, the nucleotides of this coding sequence, into groups of 3. This will actually make it easier for you to study or to locate the position of any codon and also the position of any SNP. Suppose you are talking about a mutation at the position 6 in the coding sequence, so it means you are talking about 1, 2, 3, 4, 5 and 6th codon, which is GCC. I have already split the whole coding sequence into codons, and now I will teach you how can you retrieve the SNPs from ENSEMBL database. You have already done with this step, and you have selected your gene of your transcript of interest in the gene. After clicking on the transcript of interest, then see on the left side in the part protein information, there is an option given which is variant. You're supposed to click on the variants, but do remember to click on the transcript of interest, because after this the variant table which you will see will show the variants that are only belonging to this transcript. So when we will click on the variants, then just below this table of transcripts, you will be able to see a table of variants. You are seeing here various columns, like the first one is of residues, the second is variant ID, third is consequence type, and so on. The columns of our interest are residue, variant ID, consequence type, residues, again codons, and then there are various tools like SIFT, Polyfen, CAD, Revel, MetalR, and mutation assessors. These are our columns of interest. As you know, these SNPs have been identified and predicted in human population through Human Genome Project. Now let me show you how can you read these SNPs. At the position 2, position 2 means the codon number 2, and which is in our case CTG. This is CTG, and you may see here in the codons, in the column of the codons, the CTG is shown to change into TTG. Like here, a single nucleotide polymorphism is reported in this codon, which is changing C at the first position of the second codon into T. Changing the C at first position of second codon is changing into T as a result of this variation. But in residues column, you may see there is only one amino acid symbol, that is L. You might be familiar with the symbols of amino acid. If you are not, then you may download the table from Google and compare the symbols of amino acids to find out their complete name. Like L stands for leucine. So in consequence type column, you may see here that synonymous variant is written. Actually, the synonymous variant is the variant or the mutation which is silent. A silent mutation is one which is not changing the coding sequence of a gene, which is not changing the which is changing the codon, nucleic acid in the codon, but it is not having any impact on the change in the amino acid. Like In residues, you are seeing that there is only L amino acid. CTG is encoding for the L amino acid leucine, and PTG is also coding for the leucine amino acid. It means the SNP, the ID of the SNP is given here, and you have to use this ID in throughout of your manuscript in order to show this particular variant. So the SNP which is given in at position 2 or codon 2 is a synonymous variant, like it's a silent variant. It has no effect on the change on the amino acid of the protein. Now come at the position 4. There is another variant is given. Its consequence is missense. Missense variant is one which changes the amino acid in the protein, like here the position of this codon is 4. In codons column, you may see at 4th position GAC codon is present in the normal protein, in the normal gene, but due to this variant GAC is changing into AAC. We may also see it here, like at 4th position there is GAC, and due to this variant the GAC is changing into AAC, and this is not a synonymous variant like the one we have discussed before at position 2. Like it is a non-synonymous or missense variant because it is changing D into N. D stands for aspartic acid and N stands for asparagine. So we will prefer such type of variants. I will recommend you to retrieve 3 types of variants. One we have already discussed, the missense one, and second are the frameshift variants, and prefer frameshift variant which is a frameshift deletion variant, because it will be easy for us to create a case while using the missense variant or frameshift deletion variant or the stop gain variant. So we prefer usually these 3 types of variants. And third is I mentioned stop gain variant, and you may also search these variants in this table with the help of their colors. Like for missense variant, yellow color is given. From frameshift variant, purple color is given, and for stop gain variant, red color is given. In case of stop gain variant, a stop gain variant is one in which the change in nucleotide in the codon results in the formation of a stop codon at that position. Like first codon was encoding an amino acid, and the second codon is a stop codon. And in the residues type, you may see that at the position 9 in the coding sequence, there is codon GAG which is changing into TAG. Let us look at this codon 1, 2, 3, 4, 5, 6, 7, 8, 9. Here at the 9th position GAG is present, which is now changing into TAG. TAG is a stop codon, so this is a stop gained mutation. And you may see here in the residues column that the GAG is coding the E amino acid, while in the second position there is an asterisk. Asterisk shows there is no amino acid. So TAG is a stop codon, so it is encoding, it is not coding for any amino acid. And the last one which we are in which we are interested are the frameshift mutations, shift deletion mutations, like the one which is given at the 144 position. The codon is 144, and here you can see that CTG is changing into CG, like T is deleted, and in such type of mutation you can see CTG is encoding for the amino acid leucine, while CG is coding for non. X shows no amino acid. In my next lecture, I will show you how can you construct these how can you construct the cases using these variants, and also about the tools which can be used for predicting the deleteriousness of the SNPs. Thank you so much. Hello everyone, I hope you are doing well. In my previous